Okay, let's have a start. It's my great pleasure to introduce you, Jácint Tökölyi. He is working at this department. Probably most of you know him, but still the mood or the manners request me to introduce him officially. So Jácint did his PhD here in this department. It's a quite difficult topic. He was a hardcore modeler of life history decisions, and he worked on comparative analysis of life history decisions. And after that, he moved on in a more simpler system. His opinion might differ on this. On hydras, but he still continued with life history.
Thanks. So you might know me, but this is a secret side of my life, one I'm going to talk about today. And it's about interactions between animals and microbes. I'm going to talk about Hydra, of course, but first I would like to start with an introduction on these interactions. So, of course, microbes are everywhere, and we know that quite clearly because we are affected by lots of pathogens. But it's basically due to the advent of next generation sequencing that we really realize that microbes are so abundant within and in association with multicellular organisms. So just to give you some fun facts about microbes and animals, this is a sponge, and sponges, of course, they are exposed to the environment, and they are filter feeders, and they have lots of microbes that live within the sponge body in the intercellular spaces, bacteria, protozoans, viruses, etc. But the amazing thing is that if you weigh the microbes and the sponge cells in the sponge organism, up to 35% of the sponge biomass can be made up of microbes. In humans, we also have estimates. There, the biomass is not as large. It was estimated to be around 2 kilograms of microbes, which is basically mostly your gut flora. But this was re-evaluated, and now the estimate is around a few hundred grams. But if you count bacterial cells or microbial cells, most of these are bacteria, you will find that almost 40 trillion cells of bacteria exist within and on our body, and actually this is more than the human cells we have. Collectively, this amounts, so these microbes have around 200 times more genes than the human genome. So it's a huge physiological complexity that is in there, and of course it's affecting us. You see these reports every day in popular news as well, that your gut flora is important, it's affecting lots of things. Of course, since most, at least in humans, most of these microbes are in the gut, they are involved in digestion, so they help us to break down complex food stuff, and also meanwhile they synthesize essential vitamins, for instance. They are also involved in immune defense, so they are sort of part of the immune system of the animal. They protect the organism from invaders simply just by occupying the space there. It's a part of the immune system. It's a really novel research area, but there are some very interesting observations suggesting that it can affect human psychology as well, regulating moods by through affecting neurotransmitter synthesis, or some of these microbes even produce neurotransmitters themselves. And various diseases are due to an imbalance of the microbiome or inefficient function of the microbiome. So it's physiologically very, very important. And I would just like to, there are lots of examples how the microbiome affects animal physiology, but I would like to give some of my personal favorite examples of microbe-induced physiological changes. I think all the interest and the huge increase in interest in microbiome research started with basically this study, or maybe a predecessor of this, when researchers took mice from the same strain, so genetically inbred, very similar mice, one of which was obese and the other was a lean mouse, and they treated them with antibiotics and obtained very similar phenotype. And also they took mice that were germ-free without any bacteria. They lived under sterile conditions, and they force-fed them basically feces from an obese mouse or a lean mouse. And what they found is that you can basically make a mouse, a sterile mouse, obese if you feed them microbiome from an obese mouse. And amazingly, this has been even reported in humans, human twins. If you find a lean and an obese part of the twin, if you take a fecal sample from these twins and you feed them to germ-free mice, then the mouse receiving a fecal source of microbiome from the obese person will get obese themselves. So this shows that there is a causal effect of microbiome and obesity and metabolic syndromes. But this is just the starting list. If we go further, nowadays there are many other studies that show that the microbiome is not only involved in metabolism and obesity, but also regulates development itself. For instance, this very nice study done on jellyfish, scyphozoan jellyfish, which have a complex life cycle. They start with the polyp stage, and then they have this phenomenon called strobination, where the polyp produces these ephirae, and the ephirae transform into the jellyfish. And it has been shown that if you remove bacteria from these polyps, then this whole process of strobination and epheral production will be delayed. It's much slower. The jellyfish that are produced in this way are smaller, and they have an altered feeding capacity. Basically, the nematocytes decrease in abundance, so they are unable to feed. So in this case, it was hypothesized that it's the presence of cyanobacteria within the microbiome of the jellyfish, the polyp, that produces or stimulates the production of a compound called retinoic acid, which is involved in intracellular signaling in jellyfish. So if you remove the cyanobacteria, this intracellular signaling will be perturbed, and there is no or an altered developmental pattern. A very similar developmental pattern, which personally I like very much, is regeneration, which in flatworms has been shown to be affected by the presence of microbes. And if you have a normal microbiome, then these flatworms are able to regenerate. But if you perturb that microbiome, for instance, you select specific bacteria, like this Apitalia bacterium, and you put only that one, there are specific bacteria that can inhibit regeneration, and so if there's a perturbed change in microbiome composition, then regeneration can be negatively affected. And even behavior is involved. Really nice study 2 years ago on rats has shown that social hierarchies and dominance is determined by the microbiome. If the study started by describing microbiome composition of dominant and subordinate rats, and they found a difference. And they treated the rats with antibiotics, and the difference disappeared. And then they fed, force-fed feces from dominant rat to a subordinate rat, and it became dominant. So basically you can eat shit and become dominant. And another, this is just a correlative, correlative study, which is sort of interesting and emerging link, but there's no clear causal effect. It suggests that the microbiome differences might be responsible for sleep efficiency in humans. So people that have an unpredictable sleep behavior, they have an altered microbiome composition. So all this, we have a complex microbiome, and they are involved in animal physiology. So all this has led to the emergence of the holobiont concept, which means that there is no individual animal or plant as such, because the genome of the animal, along with all the genomes of viruses, bacteria, protists, fungi, they form together a common entity, which is the holobiont. And so this holobiont is increasingly well described, and interactions within the microbes associated with animals and between the animal and the microbes are increasingly well understood, and it's a really interesting topic. But today I would like to draw attention on another fact, and I would like to talk about this in continuation, and that's the fact that if you take this system, the animal and its associated microbes, this is not separated from the environment. Actually, what is a microbiome, a host-associated microbiome, is a fluid thing. It changes from time to time, seasons, years, just randomly. So all of this is surrounded by a big soup of environment and environmental microbes. And if you experience changes here, then those changes could be reverberated to the animal itself. So while this interaction is increasingly being described, we know much less about how environmental microbiota affects host-associated microbiota or animal physiology directly, whether there is a direct effect or not. But there are examples suggesting that it is important, and the environment is important in this system. Maybe the most well-known example, this really cute bobtail squid, which has a symbiotic association with a bacterium called Vibrio fischeri. They live in the Pacific Ocean, and the squid has a light organ with Vibrio, which produces light. And the squid uses this light for counter shading. So during the day, when there is sunlight, normally if you have a non-luminescent organism, it produces a shade, which predators can use to find a prey. But if you are luminescent, then you can remove this shade, basically, so predators from below you, they won't see you. So these squids use Vibrio for bioluminescence, and they acquire these bacteria from the environment. So it's not vertically inherited, but it's horizontal. When a baby squid is born, it takes up bacteria, and they grow inside. It's very specific, so only these bacteria go into the light organ. They grow inside, and there's a very interesting pattern of diel regulation. So bacteria grow during the day, and then they function during the night. And then the squid expels 90% of the bacteria into the environment. So those bacteria live there in the environment, and other squids can take it up. So in this case, normal function of the squid depends entirely on the environmental bacteria, and if you keep the squid in an environment that has no Vibrio, then it's not able to accumulate these bacteria, and it's not able to function normally. Another recent study also associated with sleep. I have many sleep examples because lots of people seem to be have problems with sleep. So at least in Drosophila, it seems to be associated with microbes, and specifically with environmental microbes. If in this study researchers have kept or have grown Drosophila in two distinct environmental samples, and they accumulated different kinds of bacteria, and what they found that taking up bacteria from one side resulted in a different sleep pattern, but only daytime sleep pattern. So night sleep was the same, but siesta pattern differed. And then, of course, this is very well known to us because it affects us humans. We have changed our environment such that we have much fewer bacteria and microbes living with us. So it is hypothesized that due to this reduced exposure to microbes, our immune system behaves differently. So we have a lack of proper immune maturation that has negative effects, and asthma and allergy is considered the result of this lack of exposure to environmental bacteria. This is the so-called hygiene hypothesis, which is mostly studied in humans. And then coming to a very nice series of studies that have been done on tadpoles, frog tadpoles. This is a series of studies done in the US where they have kept, so along this sterile environment effect, they have grown tadpoles in a sterilized environment. So they autoclaved pond water in which the tadpoles were growing, and they left the tadpoles to grow without exposure to environmental bacteria. And there's a whole series of studies with this basic experimental setup. And what they found is that lots of things, physiological patterns are affected. For instance, frogs that have grown up in a sterile environment or a reduced microbial diversity have increased susceptibility to a parasitic exposure, but they also have reduced heat tolerance, and they also can develop developmental abnormalities. So just not having bacteria in the environment is causing these physiological deviations. And it's not quite clear what is the mechanism behind all these phenomena or observations, but it can be either an indirect effect, like in the hygiene hypothesis, that without environmental bacteria there is no proper immune maturation, but it also can be so that's a direct effect, or also it can be an indirect effect when environmental bacteria colonize the host, and those bacteria that colonize the host affect the multicellular organism itself. So we found this a really interesting system, and we wanted to get some more insight into this question. Basically because there are very few studies that have looked at how sterile environments affects animal physiology, and of course, we worked with Hydra. Before presenting the experiment itself, I would just like to give you a brief description of Hydra. It's a very simple holobiont, so they are also colonized by microbes, of course. Simple meaning that they have it's just tube with two cell layers, an ectoderm and an endoderm, so there are no specific organs. The whole system is surrounded by a complex polycarbonate and polysaccharide structure called the glycocalyx. And this glycocalyx is home to the bacteria that live in association with Hydra. So there is a core microbiome, a set of species that are relatively strongly associated with, predictably associated with Hydra. It's known from quite a long time ago that this microbiome is species specific. So if you compare two distinct Hydra species, they have different microbiome in the wild, and they maintain those differences in the lab as well. And we have been working for a few years to just describe that microbial diversity. We have sampled Hydra and looked at microbiome composition in many Hungarian populations. We know that these species differences exist in Hungary as well, and we have started to describe the network of core microbes that are associated with Hydra in natural populations. So it's not just a huge diversity, but these microbes are also are known to influence host physiology, Hydra physiology. For instance, removing bacteria are involved in the immune system, because if you remove these bacteria with antibiotic treatment, then you get fungal development. So of course you remove the bacteria, there is an empty niche there, and the fungi can just start to grow. It's involved in movement and sort of behavior, these peristaltic movements that are present normally in Hydra. They stop this if their microbiome is removed. So specific bacteria elicit some kinds of chemicals that induce Hydra to do these movements. And also perturbation of the microbiome can give rise to tumors in Hydra. So this is a very interesting case of bacteria-induced tumor development. So this is the system that we're working with, and what we wanted to find out is how interaction between the environment and the host-associated microbiome affects fitness and health status of Hydra. We did a very simple experiment. We went out to a lake, we collected Hydra, we collected water from the lake, and we followed Hydra population size basically under sterile conditions. So they had the same bacteria that they had in the lake, and we didn't open them in the lab. So there is no lab exposure here. We put them into jars with a fixed population size, and we let them reproduce there normally and followed population size through time. And we had, in this early experiment, we had 4 distinct treatment conditions. One was to keep them in their normal environment, so normal lake water. Another one was to keep them in Hydra medium. This is a synthetic medium that we use in the lab. It lacks nutrients, at least bacterial nutrients, but it's just a smooth salt solution. And we had 2 other groups, both of which were lake water that was sterilized. One of the treatments was autoclaving, so you heat it up to 120 degrees, and the other was sterile filtering. And these two differ a bit because sterile filtering removes bacteria, so it's a 0.2 micron sized filter. It removes most bacteria, but it doesn't remove viruses, for instance. Autoclaving kills everything, but it also changes the pH and salts and everything. So it's not just sterilization that happens here, but there are lots of other potential chemical changes. We need to keep that in mind when interpreting results. Anyway, what we found is that this is blue is lake water, and black is autoclaved lake water. Green is sterile filtered lake water, and red is hydromedium. So what we found is that there is a huge difference in population size when hydras are grown in their own lake water, or when this lake water is sterilized. So they basically die in a sterile environment. We repeated this experiment because I always make this mistake. We repeated this experiment a few months later to see whether it's repeatable or not. And of course, it was not repeatable. So the same experimental setup, and in this case, 3 months later, if you do the same, you see that autoclaved lake water, they still die. But interestingly, the normal lake water polyps, they also die. So there is a strange interaction between a repeatability problem here. But what I've learned from my previous experience with repeatability is that if you try to repeat something and you don't get the same result despite your best interest, then there is something biological pattern going on there behind, which you just don't understand. So, third time, we took polyps from the lake at the same time, and because these two, they were collected at a 3 months difference, but also there was a different temperature, and we maintained the temperature difference in the lab. We just thought maybe it's the temperature. So we split the group, or our polyps, into 2 groups, 8 degrees and 12 degrees. And in this case, we didn't have enough polyps to do sterile filtering, so we just had autoclaved lake water as a sterile group, and normal lake water as a normal group. And back here you have on 8 degrees, you get the difference, and on 12 degrees, you don't get the difference. So the sterile environment affects hydra population dynamics negatively, but in a temperature-dependent way. So if temperature is higher, then the effect disappears. But interestingly, it's not because autoclaved, so polyps in the sterile environment survive better. It's because polyps in a normal environment survive worse. They have a lower population growth rate. I have no idea why that's the case, but at least we know it's not most likely it's not an artifact. We have repeated this experiment as well a few other times, so it seems to be a robust pattern, at least for polyps collected from this population. So we also wanted to get some insight into what's happening there. And we did microbiome analysis. We collected polyps and we collected water samples. So we were able to compare the microbiome composition of water and polyps in different environments. And so I will try to walk you through these graphs. Basically, you have 2 distinct graphs here because these are distinct diversity measures. So Gray-Curtis index is a measurement that takes microbial abundance into account, and Jacquard is just presence-absence. It's a number version. And this is beta diversity, so it shows how similar samples are to each other. And gray is starting condition, so when we collected. and the colors are 4 weeks later when we concluded the experiment. And if you look at water samples, what you see is that the starting condition and the condition 4 weeks later is quite similar, so the water seemed the same. We have a difference in autoclaved lake water and sterile hydromedium. And you might get surprised here because we autoclaved it, so how do we get bacteria in there? Apparently, so this is a result basically, apparently yes, we autoclaved, sterilized the water, but bacteria from the hydra, which are not sterile, can go into the water, establish populations there, and reproduce and maintain bacterial population in this sterile environment, but that's different from the original one. So other bacteria will end up in the water. And if we look at polyps, what we see is that indeed we see a difference between normal polyps cultured in a normal lake water and polyps cultured in an autoclaved environment, which is sort of what we would expect if there's a change in microbiome due to the sterile environment and that's affecting the hydra population dynamics. But sadly, we didn't really detect a temperature difference. So what we see here in a difference between temperatures is not really replicated in the microbiome data. So we cannot really say whether it's really the change in the microbiome that is causing these differences. We also looked at which bacterial species change in abundance, and again, if we look at polyps themselves, we see that some bacteria change in abundance on the surface of the polyps if they are cultured in a different environment. So in this graph, this is log-fold change. So negative values mean that that bacterium was reduced in abundance, and positive values means that it increased in abundance. And the black dots shows comparison between autoclaved lake water and normal lake water. And the red dot shows a comparison between hydromedium and normal lake water. So what we would expect is that because polyps in the autoclaved lake water, so sterile lake water, they had a lower population growth, we would expect that some bacteria maybe accumulate on them and they die because of that. But it's apparently not the case. We detected the same pattern in both hydromedium and sterile lake water. If we look at water, we see some potential candidates, like this unknown Flavobacterium, which increases in sterile lake water but doesn't increase in hydromedium. Although they do the same both on 8 and 12 degrees, so it's really far short to say that they could be causally involved. Anyways, we were excited by these unknown Flavobacteriaceae, so we tried to isolate them, and we managed to do it. It's most likely a new species for science. It's closely associated with a Flavobacterium that was isolated in Turkey from fish eggs. And this is really good for us, or interesting, potentially interesting, because these Flavobacteria are known fish pathogens, and they are actually one of the most important pathogens of fishes. So maybe there was an accumulation of these potentially pathogenic bacteria in the sterile environment that could have caused these differences. So we have this bacterium, we have complete genome sequence for it, and we are in the process of finding out whether it's really pathogenic on Hydra as well, or just a coincidence. And then finally, we looked at this is a reverse approach. So previously we went top down from treatment to bacteria, and now we go bottom up. We start with bacteria and look at which bacteria could predict Hydra population size, population dynamics, and actually there are not many. This is of course needle in the haystack. You take Hydra population size in a jar and you look at abundance of a few hundred bacteria on polyps isolated from those jars, and you do a correlation. You will get lots of false positives. You try some p-adjustment method and you get something, but whether it's true or not, you cannot really say. But we found some taxa that were either positively or negatively correlated with Hydra fitness, so to say. And maybe the most interesting in this respect is Polynucleobacter, which showed a temperature-dependent abundance. So they had lower abundance on 12 degrees and higher abundance on 8 degrees. There seems to be a correlation between Hydra population size and Polynucleobacter abundance, which is interesting to us because it's one of the main Hydra colonizers. It's also known to be vertically transmitted through Hydra eggs, so it's most likely a symbiont, because the Hydra, they don't really let any bacteria go into the eggs. So if it's allowed into the egg, they regulate it with antibiotic peptides. If they let someone into the eggs, then that's probably a useful species. So this is a potential candidate bacterium that can predict Hydra population size. And so with this, we have lots of questions. We don't really, there seems to be a pattern that culturing Hydra in a sterile environment has negative effects on their fitness. It's repeatable, at least in one population. We don't really know what could be the explanation. We didn't really see a difference in microbiome composition between polyps that were cultured in different environments. But of course, this microbiome sequencing is just a raw estimate of a composition. So you could have some taxa that are low in abundance but have a huge effect on the polyp. So we are suspecting that maybe it's the dysbiosis or the change in the microbiome composition of Hydra that could have caused these effects, or we suspect that the presence of these pathogenic bacteria could be involved as well. We need to track them, to learn to track them more precisely, and to experimentally manipulate this system with the pathogen. We have them in an agar plate, so we can just sprinkle hydras with pathogens and kill them. That's easy if they die. But of course it can be something else. So there are lots of questions. We weren't really able to understand the mechanism, but at least it seems to be a distinct mechanism than the ones that have been described previously. It's most likely not an immune maturation thing happening in Hydra, because they don't have an adaptive immune system. Also, it's most likely not an indirect effect through host colonization. There could be something else that is affecting it. So with that, I'm finishing. This was basically a study done by Mate and in collaboration with the Metagenomics Institute from Debrecen and Sebastian Fraune from Düsseldorf. And thank you for everyone, and thanks for your attention.
I
finished for you.
Yeah, I need to go, but I'll just see you later. Yeah, very well done, very exciting. Okay. Thank you, Jácint.
Thanks.
Thank you very much. Questions? No one has one? I have one. Go ahead.
You don't want to start?
No.
I was curious about this, the temperatures of water. So it seems to be really like a narrow range. How does that compare to the natural seasonality of water temperatures, or what the hydros see?
So the temperatures that we selected were similar to the conditions during sampling. So we wanted to maintain similar conditions in the lab as they experience in the field. So these are just 2 degrees, which are narrow range, because at the higher temperature it is difficult to collect hydra in large abundance. These are psychrophilic species. They love the cold, and if it's the temperature, the summer is coming and the temperature is high, then hydra abundance decreases. So we are struggling with finding enough individuals for an experiment. But still, already this 4 degrees had an effect. So of course, it would be really interesting to see whether the same effects hold on a wider temperature range. We need to explain the 4 degrees effects first.
So you guys didn't look at regeneration or anything in relation to sterilization?
No, that would be an interesting additional. So because we looked only at one fitness measure, that's population size, which is easy, and we didn't try additional measures of health and fitness. Partly because it's more difficult because you need to keep them sterile and you need to work under a sterile conditions. So if you just put them in a jar, you can follow them and you don't need to worry about introducing lab microbes into the system. But it would be interesting to have a look at that.
I know that species delimitations can be somewhat problematic in the microbes, but can you say something about how specific are these strains of bacteria that you find in the hydras? So can you find it in other hydra species? Can you also find it in the flatworms or maybe even in vertebrates?
Some of them, yes, are associated with other species. Most of so at least this microbiome analysis was narrowed down on genus level, because the type of sequencing that we use doesn't really allow species detection. And of course, at the genus level, the taxa that are in the hydra microbiome, Rhodophyrax, Aeromonas, Flavobacterium, they are everywhere. They are in the microbiome of most other animal species. Polynucleobacter, which I briefly mentioned here, is another interesting species in this matter, because some Polynucleobacter species or subspecies, they are intracellular symbionts of ciliates. We did a phylogenetic analysis on this Polynucleobacter, and apparently our samples are full with different kinds of Polynucleobacter. Some of them are free living, others are associated with ciliates. So this is just the surface, of course, and probably if we would look at the species level or more specific identification, then we could get a different picture.
Thank you.
What did you feed the hydra?
We didn't feed these.
So they just leave there. So it's not surprising that their population is crashed.
At the end, they starve to death. Yes.
And how can that influence the results? Because you are studying a dying population, dying individuals, basically.
Yeah. So if, of course, if you have a well-fed animal, then all this relationship could change. So the nutrient level is probably hugely important. It's just it's much easier to do it without feeding animals. And again, if you feed them, you have this problem with introducing lab bacteria. So these results are specific to dying hydra in stress, and yeah, that's specific to that condition.
And what you might argue that under the stressful conditions, the microbiome might be more important.
Yeah, could be.
Yeah. Other questions?
Well, I have one, just realizing this and in relation to this. So it should matter then the hydra that you randomly assign to these groups, how much reserves they have, right? So how large they are and how much capacity they have to cope with this.
Yes.
Can you randomize this or?
Yeah, they were randomized. So each of, I didn't mention this, each of the jars we had, and each of these lines is the mean of 5 distinct jars, and they had a starting population of 10 individuals. So it's sort of, it's not a single individual, but you have 10 individuals, and they can grow quite a lot, because already when we bring them into the lab, they have buds, and they release those buds, and for a few days they can still continue to produce buds from their reserves. But it should be the same in each treatment group, because we didn't specifically select for.
Did you measure the composition of the waters, the different type of waters? I mean, the chemical level.
Yes, we had a look on that, and of course autoclave lake water is different, because if you autoclave water, all the living things in there will die and they will start to decompose, but also salts will precipitate. So it changes pH, it changes the electric capacity of the water, so lots of things.
So is it possible that autoclaving destroys some nutrients for the hydra?
Could be. But as far as we know, they don't have any essential nutrients that they take up from water. Maybe there are some vitamins in there that could be affected. So that's definitely a potential explanation. Although we think, since we detected a difference with sterile filtering alone. The effect was not as large, but still, no, not this one, it's the 8 degrees. So the sterile filtering had a similar difference, although not as large as autoclaving. But another interesting question is why sterile filtering and autoclaving have different effects. We don't really know that. So it started as a simple experiment, but
Life is complex.
life is complex.
Can it be the case that autoclaving produces some kind of poison for the hydra because of this complex chemical reactions going on during the sterilization?
Yeah, if you have salt accumulation, that could be an issue. We were struggling to find a better method to sterilize water. So we also talked about cyanobacteria producing toxins in there. So or not producing enough oxygen. So we also tried to keep. We had an experiment where we had a group kept in dark to kill just specifically cyanobacteria, but that one had the same effect, so apparently it's not that, but I wouldn't put my life on it.
Other questions? Thank you again.